Epigallocatechin 3 Gallate decreases VEGF production in head and neck and breast carcinoma cells by inhibiting EGFR-related pathways of signal transduction. In a recent study on head and neck squamous cell carcinoma, HNSCC, cells, we found that epigallocatechin 3 Gallate, EGCG, a major biologically active component of green tea, inhibited activation of the epidermal growth factor receptor, EGFR, and related signaling pathways. Since activation of EGFR signaling pathways is associated with angiogenesis, we examined the effects of EGCG on vascular endothelial growth factor, VEGF production by YCUH891 HNSCC and MDAMB-231 breast carcinoma cell lines. Because we found that both of these cell lines display autocrine activation of transforming growth factor alpha, TGF-alpha, EGFR signaling and produce high levels of VEGF. Treatment with EGCG inhibited the constitutive activation of the EGFR, STAT3, and ACT in both cell lines. These changes were associated with inhibition of VEGF promoter activity and cellular production of VEGF. Mechanistic studies indicated that inhibition of STAT3, but not mitogen-activated protein kinase, MEK1 or phosphatidylinositol 3 kinase, PI3K, significantly decreased VEGF promoter activity. However, the inhibitory effects of a dominant negative STAT3 on VEGF expression was not as strong as that produced by EGCG. An analysis of alternative pathways indicated that EGCG strongly inhibited the constitutive activation of NF-kappa B in both cell lines, and an NF-kappa B inhibitor strongly inhibited VEGF production. These results suggest that EGCG inhibits VEGF production by inhibiting both the constitutive activation of STAT3 and NF-kappa-B, but not extracellular signal-regulated kinase, ERK, or ACT, in these cells. Therefore, EGCG may be useful in treating HNSCC and breast carcinoma because it can exert both antiproliferative and anti-angiogenic activities.